Hello, everyone. Let's continue on with our next presentation titled Advancing Process Efficiency for the End-to-End Manufacturing of T-Cell Immunotherapies. This webinar is presented by Drs. Paula Lam and Sylvie Shen. Click the Speaker Information tab on the left of your screen for a complete biography on our speakers. Again, we encourage you to submit questions during the presentation, and we will answer as many as we have time for afterward. Dr. Lam, Dr. Shen, the floor is yours.
Thank you for attending today's webinar. My name is Sylvie. I'm an application scientist supporting cell and gene therapy at Thermo Fisher Scientific. For today's talk, I will walk through Thermo Fisher's approach to the end-to-end manufacturing of adoptive T-cell immunotherapy. We also have the honor of having Dr. Paula Lang, Chief Scientific Officer of BioCell Innovation, co-present her efforts on developing a humanized GMP lentiviral CAR construct. To start, we will discuss how Thermo Fisher's innovative technologies could overcome manufacturing hurdles for cell and gene therapy. We will present our process following an efficient and closed 7-day viral-based CAR-T production process using BioCell GMP lentivirus construct. Finally, we will share some insights on a recent 24-hour rapid CAR-T manufacturing process developed by our R&D scientists. While the field of cell and gene therapy is rapidly progressing, numerous challenges remain in the realm of manufacturing. One key challenge is how to provide patients with safe, efficient, and potent cellular therapies that are also affordable and can be delivered in a timely manner. As depicted in the workflow on the right, cell manufacturing involves multiple steps that begins with cell isolation and activation, followed by gene modification using either viral or non-viral method. Subsequently, the cells are expanded in bioreactor to obtain larger outputs. The process continues with harvest, formulation, and cryopreservation of the final product. And finally, the product undergoes characterization and release testing before patient infusion. Cell manufacturing is a complex process characterized by many open and labor-intensive steps that are susceptible to contamination risks and also prone to error. Some instruments lack the flexibility and are unable to accommodate different applications and protocol modifications. Process tends to be lengthy, require extensive quality control steps, resulting in high costs. With that in mind, Thermo Fisher has developed closed system for cell processing and with validated protocols using GMP-compliant raw materials to address many of the challenges mentioned earlier. Closing the process could allow manufacturing to be conducted in a GMP Class C environment. As shown in this diagram, our closed system supports each step of the manufacturing workflow, starting from magnetic isolation and activation of T cells directly from leukapheresis products to the expansion in common closed culture vessels, followed by gene modification and final formulation. This slides features our complete suite of solutions to enable closed modular manufacturing of gene-modified cell therapies. Starting from the left, the GIPCO CTS DynaSelect is a closed, automated, and column-free magnetic separator for the isolation and activation of T cells. Isolated T cells would be seeded in a closed culture system of user's choice. The CTS Roteum counterflow centrifugation system would then be applied for closed cell washing and buffer exchange across multiple steps. To engineer cells using viral-based approaches, the users would simply supply the virus to the closed culture vessel or bioreactor. For non-viral-based gene engineering, the CTS Xenon is a closed large-scale electroporator that could electroporate cells in up to 25 ml of volume. And finally, Thermo Fisher provides a wide array of benchtop analytical solutions and assays for lot release and safety testing. The key advantage of modularity is that it allows the flexibility in process development and supports cost-efficient scale-up and scale-out. Each closed system could be used as a standalone instrument to support each step of the workflow. This modularity allows the segregation of the most time-consuming steps of the workflow, which is the cell expansion step, allowing the users to free up the use of other instruments within the workflow. Another key advantage of our modular system is the digital connectivity. For each instrument inside the workflow, it can function as a standalone device. It can also be digitally integrated into a complete workflow through our new software solution. The CTS Cellmation allows operators to connect and control cell therapy instruments offered by Thermo Fisher. The software is powered by Emerson's Delta V distributed control system and provides a flexible platform for integrating multiple systems. Overall, the CTS Cellmation software is optimized for a smooth transition from process development through to clinical trial and the commercialization. Next, I would like to provide more insights in our closed system and the GMP compliant reagents for CAR-T production. This slide introduces the CTS DynaSelect magnetic separation system, which closes and automates the T-cell separation process. DynaSelect was designed to be used with CTS DynaBeads that is well established in the field and has already been applied in numerous clinical applications. A key feature that I would like to highlight for DynaSelect is the scalability. DynaSelect supports both autologous and allogeneic workflow. It allows continuous process of up to 10 liters of sample volume. The system also consists of an intuitive interface that empowers users to customize their protocol for cell isolation, activation, and depletion. We have optimized the T-cell isolation directly from frozen or fresh leukopak without need for washing or PBMC isolation. We leveraged our CTS CD3 CD28 DynaBeads, which combines the isolation and activation in one step to do so. Here we show high purity of isolated T cells using the DynaSelect. In addition to isolation and activation, Thermo Fisher has developed a range of GMP-compliant CTS reagents for T-cell expansion and gene modification. We offer a complete system for suspension-based lentiviral production and the relevant tools such as GMP Cas9 protein for gene editing. For robust T-cell expansion, our renowned serum-free and xenon-free medium is the CTS Optimizer system, and supplemented with our GMP PepperTech cytokines. A customized format such as packaging or size for these specialized media are available upon request. And more critically, our reagents are developed and optimized to selectively promote the expansion of naive stem and central memory T cells. Published studies have demonstrated that the infusion of younger, less differentiated T cells correlates with higher therapeutic potency. And due to the superior engraftment, persistence, and anti-tumor immune response conveyed by these younger cells. The younger phenotype T cells exhibits higher proliferative capacity and enhanced cytokine production, while the more mature cells harbor stronger effector function. And moving on, now that another important closed system in our CAR-T production workflow is the CTS Rotea counterflow centrifugation system. As shown in this figure, this novel technology allows users to perform a variety of applications across multiple steps using a single kit format. Similar to DynaSelect, users are empowered to create and modify their protocol at no cost. And two key benefits of the Rotea counterflow technology is the separation of cells by size and achieving cell output volume as low as 5 mil. An example of how we leverage these features is to separate and remove dead cells from expanded culture. And another example of the cell size separation using Rotea is the separation of monocytes from lymphocytes. In this case study, we processed a patient apheresis sample that had higher monocyte contents of above 30%, as shown on the left. And the user wanted to deplete the monocytes due to concerns over impact on T cell recovery after magnetic isolation and expansion. After around 20 minutes of processing time, we demonstrated almost complete depletion of monocytes, 72% enrichment of lymphocytes, and a high viability of over 90%. And overall, the CTS Rotea system is very well known for its versatility and for its many advantages. And since launch, we have placed over 400 units globally, with a growing number used in clinical setting. As a wrap-up to the first session, I would like to share some insights on how Thermo Fisher collaborates to assist developers in accelerating progress. Cell and gene therapy manufacturing is complex. As such, we have put in place a global collaboration program supported by a dedicated team of scientists to share our knowledge and to optimize process for development utilizing our reagents and our platforms. Shown on these slides is a case study around our collaboration with Arsenal Bio. Arsenal Bio is a clinical stage company engineering CAR T cell therapies for solid tumor. They adopted our suite of closed systems and leveraged support from our scientists to close their process that enables them to fast-track their process development and resulted in the dosing of the first patient in early 2023. In line with the topic of collaboration, Thermo Fisher has collaborated with BioCell Innovation to evaluate their humanized GMP lentiviral construct to produce CD19 CAR T cells following an efficient closed process using CTS instruments and reagents. And with that, I'm pleased to hand over to Dr. Paula Lang, Chief Scientific Officer of BioCell, to provide more insights around BioCell's humanized lentiviral CAR construct. Over to you, Paula.
Thank you, Sylvie, for the introduction to the various Thermo Fisher technology platform. I would like to start by introducing BioCell Innovation, which is a Singapore-based startup company. Our primary objective is to bring cell and gene therapy Our primary objective is to bring cell and gene therapy to patients in need, offering them affordable and effective therapy. In today's presentation, I would like to share with you the generation and characterization of an in-house humanized CD19 CAR T vectors. Since 2017, there has been 6 CAR T cell therapies that have been approved by the Food and Drug Administration. 4 of these are targeted against CD19 and 2 are targeted against BCMA. The overall response rate ranged from 72% to 98%, but the cost is highly set at a minimum of USD 375,000 in a recent check. Emily Whitehead is the first patient receiving CAR T therapy against acute lymphoblastic leukemia in April 2012, and she has remained cancer-free until today. The specific CD19 CAR received by Emily belonged to a CD19-specific immunoglobulin subtype 1 mouse monoclonal antibody known as FMC63. The presence of non-human scFv might induce human anti-mouse antibody known as HAMA response, and thereby limiting CAR T-cell persistence in patients. There have also been case studies to show that patients with relapse or refractory acute lymphoblastic leukemia that had failed FMC63-derived CD19 CAR treatment is responsive to humanized CD19 CAR therapy. Lee and colleagues in 2021 attempted to compare the efficacy and safety of cells transduced with murine and humanized CD19 CAR, and that the latter had shown a higher rate of ex vivo proliferation and overall improved cytolytic activity, as well as persistence of the humanized clone. Although there might have been differences in the method of analysis, BioCell's humanized CD19 CAR T cells also exhibited good proliferation rate and tumor cell killing efficacy in a co-culture system, and no toxicity was detected in vivo. Thus, we constructed a second-generation CAR vector comprising of humanized anti-CD19 scFv with a CD8 transmembrane followed by 4-1BB and CD3 zeta co-stimulatory domains. The co-stimulatory activation domains were also deleted to generate a mutant construct as match control. We then generated stable clones of NAM6 and Raji cells expressing the luciferase reporter gene. NAM6 was originally derived from a patient suffering from acute lymphoblastic leukemia, while Raji were derived from a patient with Burkitt lymphoma. The result shows that only cells transduced with humanized CD19 CAR T cells exhibited significant cell kill from the PBMC control at the various effector to target ratios, where cell kill exhibited by the control vector, which is the delete construct, were insignificantly different from the PBMC control. We subsequently measured the levels of pro-inflammatory cytokines, interferon gamma, and TNF alpha when CD19 CAR T modified cells were co-cultured with either NAM6, LUC, or Raji LUC after 24 hours. The supernatant was analyzed by multiplex ELISA, and the results showed that both interferon gamma and TNF alpha were significantly elevated in both cell line models, whereas the level of cytokines secreted by cells transduced with the CD19 CAR delete was insignificantly different from untransduced cells. In view of the demand for producing more lentiviral vectors to perform clinical animal work, we partner with Cellvac, which is the first CDMO in Southeast Asia approved for providing clinical-grade lentiviral vectors. The lentiviral vectors produced by Cellvac is of high quality in accordance to PICS guideline for medicinal product and using Cellvac proprietary helper plasmids. We generally obtain 2.3 to 5.8-fold higher lentiviral vectors when compared to other commercial helper plasmid system. We also shown a 3.7-fold increase in viral titer when the same gene of interest was subcloned into Cellvac transfer vector backbone versus the client vector backbone. The focus of the upstream processing is to produce the lentiviral vectors in bulk, which typically involve co-transfection of lentiviral constructs carrying the gene of interest with the packaging helper plasmids. The downstream bioprocessing comprises of individual operational units to achieve purification and concentration via clarification, chromatography, ultrafiltration, and diafiltration steps, and subsequently reformulate to the final fill. Each of these steps must be carefully optimized as lentiviral vectors are sensitive to mechanical shear force, pH, salt, temperature, buffer osmolarity, and the entire bioprocessing of lentiviral vectors at Cellvac take 15 days to complete. The common impurities within the bioprocessing of the lentiviral vectors could originate from the lentiviral vectors as well as from the manufacturing process itself, as summarized in the blue color table. For the lentiviral vectors, there are viral aggregates, there are free vector envelopes, non-functional vectors that are either broken or immatured, as well as other vector components and proteoglycans. For the process, there are host and plasmid DNA, host cell proteins, lipids, nucleases, culture leachable, and serum-derived contaminants. In short, Cellvac lentiviral bioprocesses can remove more than 99.5% of the impurities. The orange color table represents 3 GMP production runs performed at Cellvac, showing consistently reproducible high titer by flow cytometry and qPCR analysis. The TU per P24 value estimate the overall specific activity of the lentiviral vectors, and apart from the assays to determine impurities, we also measure sterility, endotoxin, mycoplasma, and replication-competent viruses to ensure that the GMP lentiviral vectors meet the current safety requirements. Given the relevant elevated enhanced interferon gamma induced by CD19 CAR T cells in vitro as seen on the upper left-hand corner, we then perform in vivo experiment using the lentiviral vectors prepared from Cellvac. Not-skid gamma mice were sub-irradiated and injected with 1 million of NUM6 loop tumor cells. The mice were randomized into 3 groups and systemically infused with either PBS, 5 million human T cells, or 5 million of either CD19 CAR or CD19 CAR delete. The bioluminescence signals was tracked and tumor burden was quantitated at representative time point of day 21 post-infusion. Similar average body weight of the treated animals when compared to non-treated animals suggested there was no significant cytotoxicity induced by the treatment. Elevated interferon gamma was detectable in serum sample from mice treated with CD19 CAR when compared to the controls, indicating that CD19 CAR T cells are functional and potent. In conclusion, we have generated a second-generation humanized CD19 CAR, and the preliminary data have shown that these vectors effectively transduce T cells and demonstrate functionality and safety both in vitro and in vivo. The low amount of lentiviral vector use indicate a lower corresponding lentiviral production cost and simultaneously increase the biosafety associated with using lentiviral vectors. We are collaborating with Thermo Fisher Scientific in providing humanized CD19 lentiviral vectors to improve the cost productivity and efficiency of the CAR T manufacturing process. And with this, I would like to return the presentation to Sylvie, who will discuss about an end-to-end CAR T manufacturing workflow solution. Thank you.
Thank you, Paula, for providing exciting insights about your work. Now I shall discuss how we used BioCell's humanized CD19 CAR lentivirus in our validated end-to-end viral CAR T-cell production process. Our process was designed with one thing in mind, and that is to reduce the cost to patients. This is done by creating a modular workflow, increasing the efficiency and robustness and the simplification of the full process. Briefly, we perform a one-step isolation and activation of T cells directly from a thawed leukopak using CTS DynaSelect and CD3 and CD28 DynaBeads. The instrument will seed the cells directly into the closed G-Rex vessel, which is then placed in an incubator. After 24 hours, we add the lentivirus directly into the G-Rex and top up with medium. An additional supplementation with fresh IO2 was performed every 2 to 3 days. At the end of a 7-day process, expanded cells are harvested and the beads are removed on the DynaSelect. Following that, the CAR T cells were next processed on the rotator for wash, concentration, and the final formulation and cryopreservation. Samples are taken for characterization and release testing. We've used 3 different healthy donor leukopak that were commercially available to validate our 7-day process. While we had assumed that they would be similar, our flow analysis showed a significant difference between their viability and the monocytes contents. As such, our CD3 T cell recovery post-isolation was on average around 80%. And note that the lowest recovery was seen for the sample with high monocytes contents. One consideration for such sample might be to deplete these monocytes first using the rotator. However, despite this, we observed a very high level of purity of cells per T cells across all donors after 7 days of expansion, shown in the bottom left panel. This suggests that our process is robust and consistent enough to handle some variability among donors. And we also observed an average of 32-fold expansion of the T cells, which typically result in 2 to 3 billion cells in a 1-liter G-Rex vessel. As we move ahead, I will discuss some more data derived from donor 3 with the higher monocytes contents. In the left panel, you can see that the donor 3 had 40% monocytes 40% monocytes contents. However, after activation and expansion used DynaBeads and Optimizer Pro system, we observed over 98% T cell purity at day 7 of our process. Likewise, the expression of stage-specific activation markers was consistent with the literature and demonstrating the progression of the T cell activation throughout the 7-day production period. A key aspect of reducing cost is to perform optimization on a smaller scale before moving to a large scale. However, this means that the process must be scalable. And here we demonstrate that our workflow is indeed scalable. We observed comparable cell viability and the fold expansion of T cells following our 7-day process between small-scale 6-well G-Rex and the 1-liter G-Rex closed vessel. As mentioned earlier, a high percentage of central memory T cells has been associated with greater persistence and anti-tumor immune response. Our median end process resulted in an enrichment of central memory T cells post-transduction, with on day 7, the T cell population was enriched with a central memory phenotype for up to 96%. As stated earlier, one of our goals is to simplify the process. In the case of transduction, we perform the transduction without prolonged spinoculation and without the addition of any enhancers. Using BioCell's purified GMP-grade lentivirus, we observed 46% and 60% CAR-positive cells on day 7 at MOI of 5 and 10, respectively. To further optimize our process and to enhance the transduction efficiency, our R&D team adjusted several parameters, including the seeding density, seeding volume, MOI, and days of transduction. We were able to reduce the entire process from 7 to 5 days. We observed 60% CD19 CAR-positive expression at a lower MOI of 4 and a similar expression post-SAW. We harvested 1 billion cell CAR-positive T cells per liter of the culture. On the right, we conducted cytotoxicity assays against CD19 positive NAM6 cells and observed efficient and comparable killing between our 5-day and the 7-day process. Overall, our 5 to 7-day process is robust and straightforward. And Thermo Fisher is always looking to improve the process, so we welcome new collaborations and would be happy to share more details about our process with you. And finally, I would like to touch on our 24-hour rapid manufacturing process, which is an exciting area in the field today. This work has recently been presented by our R&D scientist, Dr. Mina Ahmadi, and you could view her recent recording on the BioInsights. As discussed earlier, cell manufacturing faces challenges of high cost and long production duration, particularly problematic in autologous setting. A rapid process is attractive for quicker access and reduced production costs. Studies have shown that a quicker process yields T cells product with a younger phenotype and higher potency. Notably, in the paper shown on the left, one-day manufactured non-activated T cells demonstrated increased anti-leukemic activity compared to standard protocols. Additionally, a clinical study showed efficacy in rapid production CAR T therapy for leukemic patients. And the CAR T manufacturing process can be shortened by eliminating the expansion step. And this is also enabled by the development of detachable Dynabeads technologies that enables the transformation of a standard 7-day process to a 24-hour rapid process. And very briefly, the data on the left shows a high percentage of T cell purity in the 24-hour process product comparable to what's been observed in a 7-day process cell product. On the right, we observe a higher percentage of naive T cells in a 24-hour process and more central memory T cells from a standard 7-day process. We also demonstrated that 24-hour product showed improved expansion capacity and cytotoxicity compared to the CAR T cell manufactured using a standard 7-day process. If you like more detailed information, you can view Dr. Mina's webinar on bioinsights for this 24-hour rapid production process. Just to summarize, we've demonstrated an efficient, closed, GMP-compliant CAR T production process that was with 24 hours, 5 days, and a 7 days process. All process showed high enrichment of T-naive and T-stem central memory populations, and shows all the process showed high purity and high transduction efficiency, and robust cytotoxicity was shown with BioCell's GMP CD19 CAR lentiviral construct. The studies are ongoing to assess more donors and for safety testings. And lastly, collaboration and partnering in the cell and gene therapy field are required to accelerate the progress in the field. And we like to thank our R&D scientists for their contribution and work on the project, and also thank you for BioCell for supplying the GMP lentivirus. Thank you.
Well, thank you both for that informative presentation. We will now start the live Q&A portion of this webinar. Again, to our audience, if you have any question you'd like to ask, please do so now. So let's get started. We have a question in from the audience I'd like to start with. Say, hi, thank you for the great presentation. May I know if there is only one sample allowed to be processed at one time for each of these platforms? Thank you.
Okay, I would like to take this question. Yeah, it's a good question, and I think the short answer is yes and no. As we talked about earlier, our cell therapy system is a modular system. For each individual instrumentation within that workflow, each individual instrument, then that's right, you process one sample at a time. And then comes the flexibility that offers by a modular system, which is enables the segregation of each steps. And so that for the whole system, you can, like especially the segregation of the most time-consuming expansion part, so you will be able to have more than one sample for the whole system that or in the say expansion period. For example, if you segregate out the expansion and put your cells into the expansion period, and then you free up all instrument for use for another sample. Yeah, so did that answer your question?
I would say it does. We have time for just one more question here from the audience. Can rapid manufacturing be applied to allogenic CAR T production?
Okay. Okay. Yeah, I like to answer this question as well. So rapid production, can rapid production apply to the allogeneic CAR T manufacturing? I would say that the rapid production as the advanced, or on the other hand, the allogeneic setting, it's more focused on the scale up and the scale out. So you will be able to have a economic of scale that you can apply to multiple users. And for the rapid production, once you take out the expansion period, so that in terms of scale wise, it's not on the scale up direction, and it's more suited for the autologous that's on the scale down and scale down and there's speed up, and so that you can get your product to the patient more faster and at a lower cost. And this in this sense, in the autologous setting, the scale up is less of your concern than the production time and production cost. While on the other hand, the allogeneic, if you take out the expansion step, go for rapid production, then you don't have the large enough output that to make it scalable or more scalable for more patients. That's what I think. Yeah.
Great, thank you, Dr. Shen. Dr. Lam, would you like to add anything?
I tend to agree with Dr. Shen. I do think that for the rapid manufacturing approach, in theory, it should be applicable to the allogeneic CAR T platform as well. But I think it will require some optimization. As you know, the complexity of the entire production, there's many key parameters that need to be fine-tuned. But it's definitely something I would see feasible as off-the-shelf production once it is fully optimized.
Wonderful. Well, Dr. Shen, Dr. Lam, thank you so much for participating in the Q&A section here. Before we end this webinar, I would like to encourage everybody to take a look at that session survey found on the left side of your screen. Go ahead and take a moment to fill that out. Our next presentation, titled The Experience of Development and Verification of Analytical Platforms for Cellular and Gene Therapy, or CGT, products in EMO Biomedicine, is up next, and will begin in approximately 2 minutes. So to join that webinar, you will need to close this window, just like before. And in the auditorium, you'll just find the button with the title of that webinar. So we will see you shortly. Thank you.
Thank you.